I think we can get started.
Yeah.
So in terms of the agenda, there's not a whole lot to go through today, or there's not a whole lot that's gone on over the past month. So the summary will be pretty short. Mostly comes down to what's going to be. We'll be discussing Rebecca's work and coming up, but Stefano from Charles River is going to be giving us an update on some new data from the backup series. And then we'll go into this RCAS model that Rebecca and her team have been running now for a couple of months. And then we'll just finish off with next steps. So in terms of kind of a summary, we have sent M4K 2009 for MET ID and stability. So we should have that data probably in about three or 4 weeks. We've also sent 2009 and 2308 for time-dependent CYP inhibition. Max and I just got back from a brain cancer symposium that was in Banff, and we presented on the M4K model and our plans for IND-enabling studies and the budget. But M4K was actually quite well represented at this meeting. So Chris Jones presented on the rationale of targeting ACVR1 and showed some of our 2009 mouse model data. And David Drury from the SGC presented some of the data that I think Stefano is going to be discussing, along with his capabilities for doing nanoBRET and those contributions to the M4K project. And finally, Jerome Fortin, who some of you may remember was involved in the program back in about 2020. He presented some work on, he's developing a new ACVR1 mouse model. It actually carries not only an ACVR1 mutation, a K27M mutation, as well as a third mutation, PIK3 kinase mutation. So he's interested in testing additional M4K compounds in his model, and so he presented that model and plans to test right now 2163, but we can certainly have him look at some additional models. He's quite interested in that. So with that, I think I can probably stop sharing my slides, and Stefano, you will be able to share your screen.
The slides.
I don't think we can hear you.
Can you see the slides?
Yeah, now we can hear you.
Okay. Yep. Okay, trying to go full screen. Okay. Right. So we said last time we sent 66 compounds to the UNC for a nanoBRET assay after we managed to screen all the compounds in our internal ADP GLOW biochemical assays. So the best compounds, so all those with IC50 below 50 nanomolars, they were sent to UNC for the screen. And they were run on a 384 well plate format. The top 16 compounds were screened in duplicate in the previously established 96 well format with a repeat of the lead 2009. And at the same time, they also screened ALK5 for as a counter screen. And all the compounds maintain a good level of potency, and they were all more potent than the lead compound, and also quite selective with ALK5. So I put all the UNC slides and data in the appendix, and I will share slides later. So in this plot, I put together the nanoBRET assay IC50 versus the IDP GLOW IC50. And as expected, there is some drop off in the cellular assay, but some of the compounds were still pretty potent in both of them. the assay. There is a wider spread in the cellular assay as well. And in the next slide, I'm going through the SAR of these compounds. So I divided in the SAR in the groups, changing on the 4 position of this pyridine. And in particular, the 4 cyanopyridines, they seem to retain much of the potency in the cellular assay and have the reduced drop-off in the nanoBRET assay. So in particular, compounds 3300 is the overall best of the set, with a biochemical potency that matches the lead compound 2009, but with an improved cellular potency, which was confirmed in the repeat assay. Solubility is still in a good position, and the other advantage of this compound was an improved MDR1 effective efflux ratio compared to the 2009, which was also confirmed by the BCRP effective efflux ratio, which is reduced as opposed to 2009. So generally, this is probably the best compounds of the set, and ideally we would be a candidate for progression to PK to see an improvement, maybe in increased receptor occupancy in vivo, given the higher potency. The other good compounds that came up as most potent in the cellular assay was 3268 here, which is improved over the biochemical assay, but we still need to collect some ADME data for this compound before drawing other conclusions. For the other compounds with the 4 cyanopyridine analogs, many of them showed reduced potency, but in particular they suffer from higher MDR1 efflux ratio, in particular the ones with the uncapped NH on the right hand side. Moving to the 4-methyl analogs, they are closer analogs to 2009. These all are slightly less potent than the 4 cyanopyridine, with the best compounds shown here, but they all suffer for a poor ADME profile, in particular with very high efflux, which don't seem to give an advantage over the original lead 2009. And finally, then we were screening also some of the other cores, where we tried to remove the 4 substitutions on the pyridine and putting the substituents on the near aromatic ring. All of the compounds that we screen, they have a large drop-off in cellular assay, and they didn't show much of activity apart from 3278, which retained some good cellular potency, but permeability values are much worse than the original lead. So just to summarize what has been done, the 66 compounds selected to be the most potent from the IDP Glow biochemical assay were screened at the UNC in the nanoBRET assay, and 27 of them were more potent than the lead compound in this assay, and the 16 best compounds were retested in the previously established 96-well format, and they all maintain very good potency and also good selectivity over ALK5. 81 compounds were also tested in the tier 1 ADME assays, and the best compounds were reported in this slide deck. So maybe as the next steps, then we will planning, if we have resources available, maybe to some tier 1 ADME in here for the most active compounds, just to complete the SAR. And maybe progress the best compounds to tier 2 ADME, particularly with the PPB or brain tissue binding, which may be more relevant. And as I said previously, 3300 is maybe the best compounds of the set, which may have improved in vivo profile over 2009. And it would be interesting to see this one in how it performs in vivo. And that's the end of this slide deck. Do you have any other questions online?
Actually, I do have a question, Stefano. Nice presentation. Yeah. Can you go back to 3310, please?
Yeah.
Yeah, 3310. The M4K 3310. It's not here. Not in this slide. I don't remember this slide. Can you go back? Yes. Yeah, this one here.
This one.
Yeah. Can you see the 30? Yeah, it looks like the potency is good. The MDCK is not bad, really, but the cellular assays were not so good. Do you know why is that?
Yeah, it may be the solubility is too low that can interfere with the cellular assay, and maybe this value is worse.
Yeah, because this is the only, I mean, this is the only hydroxy compound you have on the 6-member ring. Like, this is quite interesting that the potency went up and the MDCK is also doing well, but the cellular assays were bad.
Yeah.
Could be solubility, like you said, Ahmed. You can see most of the other ones have a basic means to solubilize them, but in this case, the nitrogen is attached. It's more aniline-like.
Okay.
So solubility could very well be an issue.
Okay. Okay. Yeah. Okay.
For the lead for the cyano compounds that are looking really good. I mean, we had some of those ones earlier on. We had liked them quite a bit, but we realized where they had an issue was against CYP. So for your nice compounds that you have for this cyano, you might want to just check them against CYP, because we found, everything for them were good, but the downfall was CYP inhibition. They were very promiscuous across all the CYP. They were very potent. So whatever good compound you have with that 4-cyano group, it might be wise to just check the CYP liabilities. And if it passes those, then you're in a good position.
So how would this activity compare with our new lead? I can never remember the number of the new compound. 20.
What, 2308?
2308, that's the thing, yeah. Sorry.
Yeah.
I may have in appendix.
Yeah,
this one.
Yeah, that's about 2 nanomolar, isn't it? In this, is it? Oh, actually, not in this slide. Sorry.
Yeah, where we previously screened this, yes, in the nanoBRET, it was around 6 nanomolar, yes, so this was seemingly more potent, but again, it's quite a high efflux.
But then it's the undemethylated one is better in the 2281 is a bit better. So I think it looks a very promising compound to me, the cyano compound. As I think we've discussed before, Methvin, there were some compounds in the cyano series that showed no CYP issues. So I think you're right that it needs to be double checked.
Yeah.
But I don't think it's an automatic assumption that there will be a CYP issue.
It's quite interesting that you don't need that basic center, as you said. So is there a way that you can, but I guess the basic center is helping in terms of your solubility. So is there a way you can introduce a weak base, or is that kind of somewhere else in the molecule to help with your properties? Because that hydroxy piperidine did look very interesting, and also the morpholine.
Morpholine wasn't active, was it?
Yeah, exactly. So
So yes, this compound.
I remember we were trying to do this some time back.
Yeah. So how was that morpholine? Remind me, Stefano. I probably should know this. The morpholine that's not been run through the nanoBRET.
So yeah, this for this compound we didn't have any more solids to send, but yes, ideally yes, it would be an interesting one.
Yeah.
We'll probably need to remake.
But you do take a solubility hit though, right?
Yeah,
we
I would
don't
imagine.
have solubility data for these compounds. I don't think.
One question I have is, do we have the 4-methyl analog of 3300?
I need to check.
I think we did make it a long time ago.
Yeah, I think
I
that
think,
looks familiar to me as well. I think it's
It looks familiar. Yeah.
no, I think we do. We just have to go back and see what that one looks like.
I think back when I was still working a long time ago. Seems a long time ago now. We made the direct comparison compounds. I'm sure they were made.
Yeah.
Yeah, I can double check that. But I'm going to share this presentation with everyone, so that there are full curves and tables and the appendix from the UNC.
So in terms of like resources for PK, are you looking for like OICR to potentially do that, or is that something that you can do it at Charles River, or where would you be looking to have that work done?
I'm not sure it's something we'll be able to do here, but
okay.
Let me check. Let me see if there's anything we can do, and I'll get back to you. I think there's probably going to be a no, but obviously I can see who I can charm.
Okay.
I think the any ADME that we want to do should be less of an issue in terms of the kind of routine stuff. So I think we'll definitely have a bit more luck there.
It's definitely worth getting those SIP results if you can find the resource to do that.
And you have how much material do you have? You don't need much.
Yeah, I need to double check, but yeah, there may be. Surely, we have the stock solution in our compound management, but I check the solids.
Yeah, I'd go with solid if you can, Stefano.
Yeah.
Okay. Any further questions for Stefano and Charles River?
No,
I
nice
really appreciate that.
Yeah.
Okay, so we're going to move on to the in vivo work. Okay, so Rebecca, I'll turn it over to you.
Thank you very much. So yeah, Deanna, unfortunately, she's in the BSU at the minute, so she can't make the meeting. So this is a lot of her work that she's been running. Just as a reminder, in case there's anybody new on the call, but probably not, this is the study that we've got running at the minute in Oren Becker's RCAS model. The model is labelled with ACALUC so that we can do BLI imaging to track the tumour over time. We've got these 4 treatment groups comparing 2308, 2281 and 2009. The reason I point this out every time is because the study is kind of an enrolment-based study. So it's not that all the mice got injected 2 months ago and then all started receiving drug at the same time. We've got like a BLI threshold that once the mice reach that threshold, then they get enrolled into the study. So this is kind of the outline, and the next slide is the one data slide that I've got to show. So this is all the BLI data that we've got for the mice in each of the treatment arms. You can see we're kind of building up the numbers of mice per treatment arm into, I think we've got between 7 and 8 in each group at the minute. The ones with the X's kind of in the brackets, they're mice that have been culled due to neurological symptoms. We haven't been plotting survival for this because some of the curves, they're only 2 or 3 weeks post-treatment. And so that kind of throws the survival plot, the kind of Kaplan-Meier plot that you used to see, and that kind of throws it out if all the mice haven't been treated for the same length of time. So that's something we're going to do towards the end of the study and share that with you. But for now, just to show that within each group, really, there are a couple of mice that haven't. The BLI kind of goes up with treatment and the mice are cold. And then there's 2 or 3 mice actually doing pretty well. Long-term treatment, we're up to day between 46 and 74 for 2009 for a few mice, and the vehicle median survival for this is 30 days. So the compound, so 2009 is one that we've run before, so that's kind of our reference. And then the two newer compounds that we've been testing for you guys, it actually looks like 2281 is performing slightly better when you're looking at the BLI, which is not what we saw with Maria's data, which I showed previously, where she looked in a DIPG patient-derived model, which was HHJD DIPG7, where we only saw a shift in the survival for 2308, I think, and not 2281. Oh, 2009.
Yeah.
Was it? Yeah, and not 2308, which she also tested. So I think this looks promising, that we've got some mice that are kind of on long-term treatment with 2281, and they look pretty similar to 2009. This is what we've got so far, and when we get a bit further down the line, so for the mice that have kind of only been on treatment for 2 to 3 weeks, and they start getting further along, be able to be plotting the survival curve a lot better, and when we've got that, we'll share that. But yes, I think definitely a good start for 2281, which is what I think you guys were interested in seeing.
I guess head-to-head, though, this looks like 2308 is kind of a bit meh. Not.
Yeah, so I think it's not having that downward effect on a BLI as the other two compounds are having. I think maybe it's keeping them stable for a bit longer. I think in the 2308, it's the purple circle and the pink triangle that are cold, and the other ones are still alive. Even that, oh no, and the blue one. Sorry, those top 3 lines, those mice have been cold, and it's the bottom green one that's still going. So not as convincing.
That's what BLI stands for.
It's bioluminescence imaging. So when we transduce the cells with a plasmid that contains acyluciferase, and then we inject I think maybe they get oral dosing of the substrate, and it means that you can the cells that are transduced with that will glow, and you can read them with an IVIS imaging software. I wish Deanna was here to answer that better. I'm pretty sure that's that's how it works. Yeah. So it's kind of something, because with these tumours, because not as much for the RCAS model actually, but most of our patient-derived models, they're very diffuse, and so it's hard to see them on MRI. They don't form solid tumours, and so we use this imaging for a lot of our models to be able to try and track tumour growth when you can't see it on MRI. So it's pretty standard for us.
Sorry if I missed this. Which of the 3 looks to be doing the best, if you can distinguish them?
Yeah, I know. I'm trying to be as careful as I can with my words. I don't want to, hedge my bets too early on. But out of comparing 2308 and 2281, I think 2281 is looking slightly better. In terms of the longer term treatment that we're able to see for some of these mice that have been on that treatment.
And compared to 2009?
So we've got a lot of data on 2009 on lots of different models, including Maria's, and that's reproducibly gives us the same kind of survival extension every time. This will probably be I don't think Maria, no, she didn't run 2281, so this will be, I don't know if you've got any in vivo. Oh, was it 2281 she ran?
Yes.
Sorry, I'm getting confused. Where she didn't see anything, and now in this model we are seeing something. So I don't know. Maybe if Jerome can, I don't know if that works in the timescale of what you guys are thinking of, but in terms of testing this in another model.
I don't think that works on the time scale, but his model is more aggressive than this one.
Yeah.
And it's a different model. You can treat juvenile pups.
Right.
Is kind of what he's looking at doing.
So yeah, I'd be a lot more in the next, I don't know, 3 to 4 weeks, when some of the mice that have just been enrolled are kind of finished or getting close to the end of their dosing schedule. I'll be a bit more confident in telling you which I prefer between 2209 and 2281. But yeah, for now, this is what we're working with.
Okay, thanks so much.
Does anyone have any further comments or questions for Rebecca?
No, this is good. I mean, as I say, I mean, this is a model I expect 228.1 to work in. So if it didn't work in this one, then 2009 was kind of my favorite after that. But you know, 2281 looked like it might still be alive. So, but let's see, like she's saying, what the experiment should be in about 2 to 3. So what's the actual end of this experiment? I mean, I know you have all the animals targeted and stuff. When do you say that the experiment is actually done? Because like, so 3 to 4 weeks, you're going to stop, or are you just going to let them continue until it's a survival thing?
Yeah, we're going to run this to the end of survival. And so a lot of these mice are living a lot longer than I was expecting them to anyway. So they're continuing dosing. We haven't stopped dosing. So these mice that are still alive will still be receiving drug, and we can do that in our license. The reason I say kind of 3 to 4 weeks is that the shorter lines on these plots, the mice were injected in June, and so based on the ones that were injected in April and May, we're expecting them to not last really more than a couple of months. So I'm hoping that within the next month, that last batch of mice that were injected will be coming towards the end of their, before they start seeing neurological symptoms. But honestly, this is, even for 2009, I mean, the median survival, we still need to see what that looks like, because there's still a couple of mice that went down pretty early. But to have mice living out to 74 days, that's almost double what the vehicle median survival is.
So I was just looking at the vehicle. So almost all of the animals in the vehicle are gone. Just one
Yep,
left?
just that one green one, yep.
So that's very good, actually. So
yep.
Okay, thank you.
That sounds really promising.
Oh, yeah.
So it's hard to, because when we, even had a look ourselves at the survival, it just shifts every time you add new mice in. Basically, just the way the plot works, it's kind of, you have to take it with a real pinch of salt, and so it's not really appropriate to show that just yet. But as soon as we've got it, we will share it.
Okay.
Yeah.
Just my understanding, the dates shown, they are the dates that you've actually enrolled into that's when they start, then the BLI reaches the...
I think that's start of drug treatment. I think so a lot of these were injected a couple of weeks before that, and then they get enrolled 2 to 3 weeks after injection. And so these were when they were stuck. Because what we were trying to look for were patterns of is it mice from one batch or mice from, because this is a different way that we're trying to set up this experiment, whether they were going down quicker or not than other batches. But actually it looks like they're kind of pretty spread out.
Can I ask a question? Maybe I've not understood the graphs correctly. Are these all the same dose, or are they different doses? Like
all the same dose. So I think these are all at 50 mg per kg.
50 mg. Okay.
Yeah, they're in different vehicles. I think it's 2308 is in. No, 2281 is in sodium citrate.
Yeah. And 2308 and 2009 are in
the methylcellulose.
I guess 2208, 2281. Obviously, it looks like you've got sort of 3 mice that it's really going down, but then you've got 3 where it's really going up. Then I guess in judging, whereas 2009, I guess you've got none. I guess the ones that are going up have stopped. They had adverse effects. So I guess I guess it's just see if you've got 3 doing the opposite of 3 doing the other thing. How do you I guess how do you make sure that it's good, that it's definitely going to work? This is my question.
So most of the time when we take in final conclusions from this data, it would be from that survival plot. And so you will obviously, for the mice that came down early, you'll get the couple of drops down at the early time point. But then if the others have a long median survival, it basically takes that curve out. And so you're not hiding the fact that there were mice within that group that died early on, but actually what's the median survival of the whole bunch of mice that you put in the experiment, and then that's what we're quoting as median survival. And so normally people do stats around that, and you'll be able to compare then the treatment arms to each other.
It looks like 2281. It looks like it's that blue one in the middle looks like that's still going. Yeah, and that's going up.
Yeah, so we'd expect that probably to be culled for neurological symptoms soon. I don't have the details on that mice, so
Oh, I
if
see.
Deanna
I
here,
guess
we could
that's
ask her.
right,
But
okay.
yeah, as soon as it's pretty robust, as it starts creeping up that BLI threshold, there's only probably about 3 to 4 weeks before the mice show the neurological symptoms, and then we would cull them.
Okay.
Yeah.
For the animal that you culled, have you is there like PK samples taken for the plasma and brain or blood and brain?
Yeah, so we haven't been taking I don't think we've been taking plasma when the mice are cold, but we do take the brain, and I think half is being, I think this came up on in the last meeting, we're doing, I think all of it was being fixed before, and now we're doing half fixed and half snap frozen, to be able to either look for biomarkers, or if we wanted to test what drug concentrations we were getting in there. Deanna mentioned a little bit about us trying to look at drug distribution across the brain, with mass spec, across the brain slices with mass spec, to see where the drug's getting in, but not necessarily doing. We've done PK up front, and we've shown that data before, and shown that with the different vehicles that we don't see any differences in the PK, like a short-term PK. It's 2 hours post-dosing, I think we did. But I don't think there's been blood taken when the mice are cold at the end of this kind of efficacy study.
I mean, the compounds, I'm guessing, is it dosed repeatedly over the time period? So yeah, it's just a case. Yeah, I'm just thinking, with the blood and the plasma, it's just a case of kind of like monitoring, like over the repeat dosing, rather than just because with one PK you get one.
So the PK that we ran was at the end of a tolerability study where the mice had been dosed 5 days on, 2 days off for 3.5 to 4 weeks. And then after the last dose, 2 hours after that, sorry, ignore my husband in the background. 2 hours after that is when the brains were taken. So it wasn't that they only just received 2 hours of drug and then the brains and plasma were taken. It was at the end of our tolerability study.
Okay, yeah, that makes sense.
Yeah.
I guess what I'd be interested in from a PK perspective is whether the kind of plasma samples or the concentrations are the same in those animals where you can see it's doing something versus the ones that have been cold.
Yeah.
I don't know whether plasma samples can be taken from the animals that are kind of still going.
Yeah.
So it'd be interesting to see if those concentrations are different from the ones that are kind of dying, because then you may get a better understanding of concentration effect, or is there something else that's driving it?
Yeah, I mean, I can definitely speak to Deanna about it. I don't know how quickly we'd be able to get those samples. We've got PK in-house that we used for the previous study. I don't know how quickly we'd be able to get that information back to you, but I mean, we could collect them and send them, providing that our license allowed for that. But I'd have to check with Deanna.
Okay, that sounds great. Thank you.
Yeah.
Quick question. Just to know, what is the vehicle you are using? Because you said there are 2 vehicles at least.
Yep.
What is the vehicle arm? Which one are you using?
So I think the vehicle actually is a mix of both.
Oh,
I think it's, yeah, and I'd have to double check with Deanna exactly what the what the purport, because this is kind of part of a larger study that we're running as well that's got a number of different vehicles. But I can check with Deanna and share that. But I think it's a mixture of the both. And I think we did run in the tolerability both vehicles separately and together, and saw that there was no tox with that. Yeah.
Yeah. That's good. Okay.
Are these dosed as solutions or are they suspensions?
Solutions or suspensions? Sorry, what's?
Sorry, so whether they're kind of clear or whether
right. So it's my job in a couple. Deanna normally prepares the drugs. It's my job in a couple of weeks to do it while she's on annual leave, and so I watched her do it yesterday. I think the 2281 in sodium citrate is clear. 2209 is clear. 2308 is slightly cloudy in the methylcellulose.
Okay, thank you.
Yeah, I mean, it's homogeneous. There's not clumps or anything, but yeah, it's not clear.
Okay, that's great. Thank you.
Okay. Thanks, Rebecca. Really appreciate this update.
No problem.
Okay. So moving on. So sort of next steps. I've just updated this IND enabling timeline. I haven't really changed any of our goals or milestones or starting dates yet, just kind of moved where we are today. So as Rebecca was saying, we'll have more data in the next 3 or 4 weeks on this efficacy study. So I'm hoping that at that point we can pick a development candidate and then start moving ahead with some of these activities. But I think one other thing that we can actually look at that may differentiate at least 2009 and 2281 is we will get this metabolite ID and stability study. And if you recall from 2281, there was some potential drawbacks, I would say, with what species we might consider for a tox study. So I think it'll be interesting to compare the results we get with 2009 back with the results we got with 2281. And part of the reason we had some questions about that, it was brought up by Divya and Mike Briggs that maybe we want to look at the time-dependent CYP inhibition. So we've sent those compounds for that as well. So like I said, I'm hoping that based on the culmination of all the data that we have, that we're getting really close. I say that every time, but I think we're getting really close to being able to choose one of these compounds to move forward with. And then once we do that, it's kind of full bore on some of those activities that I showed on the previous timeline.
I have a quick question, and maybe Mike and I think Diana can answer this. The fact that we think we're going to be targeting kids and stuff for this indication, I mean, can you comment on, like, as I said, a lot of time we devise jobs for adults, and so a lot of the metabolic information is more for adult patients. Would there be any distinct difference between metabolism for younger kids versus adult? And so, like, the data set that we're looking at from a metabolic perspective, I mean, could that be confounding if we're kind of always focusing on, treating adult? Whereas, in kids, I mean, you expect the kids to actually be metabolizing stuff faster. I mean, are there going to be differences? And so, in terms of the way we interpret the metabolism data that we're getting, is that geared towards more adult or kids indication? It doesn't matter. If my question make any sense at all.
That might have been directed at me, but I don't think I have a very good answer for it.
I was going to say, I'll take it, or else I'll jump in.
Yeah, it's a good question. It's a really good question. I haven't seen anybody present kind of a pediatric specific panel when we've been discussing other drugs, but yeah, potentially. Yeah, hand over to Liz.
I was going to say it's a difficult one because the answer is probably yes, but obviously that's very difficult to try and show. I know when human dose predictions and things like that, it's almost just adjusting for weight, so you don't take into account, from anything that I've understood, you don't take into account differences in metabolic rate between kind of children and adults. But it's maybe something that we can look into a bit more to see what's out there in the literature, see if there's anything new that's been out recently that we can get better understanding, which may help with kind of trying to get an idea of when we go into the clinic, obviously, with the kind of child population.
Mm-hmm. Yeah, because I mean, like,
I guess I would assume.
Sorry, go ahead.
I guess I would assume that, well, I mean, the hepatocyte study is just in cells, so I don't know if there would be much of a difference between the cells they use and if there was a juvenile cell line or something like that. I mean, some of that we'll tease out in the tox study for sure, because we'll have to do it in juvenile animals, but to do it kind of before we go into the tox study, I don't know if there's a way to tease that out, like Liz just said.
Yeah, I can also reach out to some of my later stage colleagues to see what experience they have in the paediatric space as well, which may shed a little bit more light. I mean, as I say, as far as I'm aware, you almost treat a child.
An interesting question.
Yeah.
Liz, as Mike, is there anything in the SimSip data?
Not in the version that we have. It will be, I think it will be in the bigger version, but we don't have access to pediatrics.
No, I mean, in response to this sort of question, I've just had a quick search and I found one article, but it looks like basically by infancy now. I don't know what infancy is, but they get up to speed pretty quickly in terms of their SIP activity. They're quite low, obviously, when they're born, but after that, as they start to get exposed to diet and environment, things start to develop. But there is variability, it says here, particularly in 3A4. So the clearance predictions may be different, and that may be why, like you said, Liz, people generally cut the doses for age based on small body weight rather than anything else, I'd have thought.
Yeah, I know I have looked at it previously, but it was a long time ago when I was looking at paracetamol and the effect that that has across different age ranges. And I think now that you've said that, it was something along those lines that actually once you reach a certain point, maybe a year, for example, you end up in a similar space, metabolic to adults.
Okay, yeah, that's interesting. Now, that's one of the things that sometimes drive me crazy about DIPG as an indication, because, like, you know, a lot of the other companies who end up going into this space always follow an adult track for their talks for some other indication, and then they just pivot to DIPG in children. But, you know, there's still a lot of questions about, in terms of the criteria we're going to use up front. I mean, you know, how does the kid, you know, metabolism and things like those factor into selection? So I just wanted to just put it out there.
Yeah, it's something we need to think about as we start looking at human dose predictions and things like that as well.
Yeah, and I think part of that is once we actually have decided on what we think is the appropriate toxicology study, to have those meetings with the regulatory bodies to ensure that we're on the right track. Any other comments or questions today? Okay, well, I think for the next team meeting, it'll be in September, so I wish everyone enjoy the rest of the summer, and we'll get back to you in the fall. But if there is, I guess, a need maybe before that, once we have this date in, I mean, that's the tentative schedule is September. But if it comes down to a development candidate selection, if people are available, I might call a meeting around that as soon as, you know, we have data available that we might be able to make that decision. Okay. Great. Enjoy the rest of your day, everyone.
Thanks, everyone.
Thanks.
Okay.
Thanks.
Thanks, Pete.
All right.